Dear children, this is an attempt to help the students of class 10 who are preparing for their board exams. This is to help you with biology. In this video, we'll go through all possible questions in each chapter. As far as CBSE board is concerned, going through your textbook is mandatory. No questions out of the textbook will be asked. So let's start with the chapter Life Processes. Yes, children, now let's start with the chapter Life Processes. So let's go through the important questions, that is the possible questions that can be asked. So please have your textbook. You might have already marked it, so we are just revising those questions. So the first question can be asked from this portion like, why are molecular movements needed for life? So molecular movements needed for life is to keep repairing and maintaining their body structure. So you know very well that science, as far as science is concerned, core points, the main point is very important. So we'll be scored only for that. So here the core point is to keep repairing and maintaining their structures. And since all these structures are made up of molecules, they must move molecules all around the time. So molecular movement is essential for life. I hope you have noted it. Now, next question. Do we need energy when we are asleep? So let me tell you one more thing. The question which I have framed will be of one way, and the questions which you get may be of a different frame. But you must understand the concept, like, do we need energy when we are at rest, or do we need energy when we are asleep? Or even you can ask, even the question can be asked like this, like, even when we are just sitting in class, do we need energy, or we need energy? Justify the statement. So the questions can be asked in that way. Whatever be the question from this part, the answer is the maintenance functions of living organisms must go on. So that is the exact answer. The maintenance function of living organisms must go on. So to carry out these maintenance jobs, breakdown or energy is required for them. So though we are asleep or we are at rest, we need energy. That's the main concept of this question. So this is question number 2. Hope you have marked. Now we are moving down. Now here comes the next question. Simple diffusion will not meet the requirements of all cells. Justify the statement. Simple diffusion will not meet the requirements of all cells. So this is in the case of multicellular organisms, okay? So this is in the case of multicellular organisms, okay? Multicellular organisms. Simple diffusion will not meet the requirements of all cells. So simple diffusion is not enough to carry out all the life processes, because the answer is here in multicellular organisms, all cells may not be in direct contact with the environment. So that is a major point. So that's the important point there. Okay. All cells may not be in direct contact with the environment. So this is a key point. So here one more question can be asked, like, if it's in the case of multicellular organisms, then what is in the case of single-celled organisms, single-celled or unicellular organisms? There, simple diffusion will satisfy their needs because they don't have any specific organs and their cells are in contact with the environment. You can see that statement here. So input, exchange of gases or removal of waste may be needed because the entire surface of the organism is in contact with the environment. So in the case of multicellular organism, that is not possible because all cells are not in contact with the environment and we have complex structures. Next. So some of the in-text questions also shall be discussed here. So the first question is the one which we discussed now. Why is diffusion insufficient to meet the oxygen requirements of multicellular organisms like humans? So I believe you understood what I said, what I meant earlier, because the question frame which I made is different from the question frame here. Here I have made it as a justification, justifying the statement. See, like, simple diffusion will not meet the requirements of all the cells. And here you can see the question is framed in a different way. Diffusion is insufficient to meet the oxygen requirement. So understanding the question is very, very important. You must understand that this question or the answer to this question is in this part. So understanding the question is very, very important. So you have to understand the concept first. Here they asked about the requirements of multicellular organisms. Will it be satisfied by diffusion? And next question, what criteria do we use to decide whether something is alive? So in this there's a bit confusion, because criteria to decide if organism is alive, usually we look for the criteria like they move or they breathe, then they grow, they're growth related. All these are the visible movements, but there are organisms which do not show this visible movement. So the right answer is the organisms which have the molecular movements are considered to be alive. And that is the reason why the viruses are still in a dilemma, like whether it is a living or a non-living thing, because they do not show any molecular movements when they are outside the host body. Now, what are outside raw materials used by an organism? So they have simply mentioned as an organism. They haven't mentioned as plant, either specified either as plants or animals. So better you mention both: plants, carbon dioxide, water and sunlight. And I've written oxygen also here, because during respiration they take oxygen. They need oxygen. And in the case of animals, food, water and oxygen. Now coming to the next question, what processes would you consider essential for maintaining life? So all the life processes which we learn in this chapter, like nutrition, respiration, transportation and excretion. So these are the processes which are required to maintain the life. And why not reproduction? Because reproduction is for the survival of the next generation, like survival of the entire species, not an individual organism. So for the existence of individual organism, these are very important, or life sustaining processes, and hence we call them as the life processes. Now we are coming to the next topic, nutrition. So from this paragraph you'll be getting quite a lot of questions. The general question can be asked from this part is like differentiate between autotrophic and heterotrophic nutrition. So when such question comes, differentiation, very simple way to answer it is writing its definition and the examples. So definition also, you can split the definition into many parts and write. So let's see what definition we get here for autotrophic organisms. So for autotrophic organisms, let's see here. Some organisms use simple food material obtained from inorganic sources in the form of carbon dioxide and water. So, or you can write like conversion of inorganic materials into organic materials. That definition is what we learn in chapter R, environment, in the case of producers. So even you can mention the same here for autotrophs. And if this is the definition for autotrophs, and what will you write for heterotrophs? Here, the organisms use, so this what I mentioned here is the definition for autotroph, first point. And this one, organisms that utilize complex substances and which have to be broken down to simpler ones before they can be used up for the upkeep and growth of the body. So it's not necessary, you can write till simpler ones. So this is the first point for heterotrophs. And so autotrophs, then second point you can write that autotrophs independently prepare their food, and heterotrophs survival depends directly or indirectly on autotrophs. You can mention that part. Autotrophs can live on their own, but heterotrophs have to depend on either directly or indirectly on autotrophs. And then you can go for the examples. Examples of autotrophs includes green plants and some bacteria. So this part you'll get in MCQ, because in MCQ when they give you as bacteria, you may get confused, like if you may start thinking is bacteria an autotroph? Yes, few bacteria are autotrophs. And now examples of heterotrophic organisms include animals and fungi. So always write the differentiation in two different columns. Mention the first point. If you are writing the first point as the definition of autotroph, you must mention the definition of the heterotroph as a complementary point. And if you're writing next one as the example, so next side also it should be an example. So this is a way to write differentiation. You must not write as paragraph. Point by point you must mention. Now coming to autotrophic nutrition. So this part can be asked as an MCQ. Mention the raw materials for photosynthesis. It is carbon dioxide and water. And these part also will get MCQs. That is internal energy reserve. Internal energy reserve. Okay. Of plants, it is starch, and animals, it is glycogen. So it's over here. Internal energy reserve. It's an often asked question, frequently asked question. Then here, one more thing we should remember, the carbohydrates which are not used immediately are stored in the form of starch. It means during photosynthesis, plant produce some soluble form that is immediately converted into the complex form where it is not dissolved. Okay, so that part is important. Now overall equation of photosynthesis, balanced equation you must write. 6 carbon dioxide, 6 molecules of water in the presence of chlorophyll and sunlight, 1 molecule of glucose, 6 molecules of oxygen and 6 molecules of water. So balanced equation is necessary there. Now this is a direct question, like events occur, mention the events or state the events or briefly explain the events occurring during the process. Process means photosynthesis. So the 3 steps. Students usually ask whether to write these 3 steps in the same order or can we shuffle. Yeah, shuffling is also fine, but absorption of light is the first point. Okay, so only after absorbing light the next points will happen. So these 3 points are very, very important. And this is a sequel question of this. So this part, next paragraph, the question can be asked in different way, but the main concept there is during daytime desert plants cannot photosynthesize because their stomata will remain closed during daytime to reduce the loss of water or to prevent transpiration. So how do they get sunlight? Because only through how do they get carbon dioxide? Because only through stomata they could take carbon dioxide. So in the absence of carbon dioxide the process will not be complete. So question can be asked in that manner, like how do desert plants photosynthesize during daytime? Or the question can be asked using the statement given here. The steps in the photosynthesis need not take place one after the other immediately. So that can also be asked. In that case, you have to mention this. And here, prepare an intermediate. So this word is the key word, okay? Prepare an intermediate means during daytime they cannot take carbon dioxide, so during night they take carbon dioxide, convert it to an intermediate, and once when light is available they'll use this intermediate, convert it to the usable form and undergo photosynthesis. Hope that's clear. So I have mentioned the question here, like how do desert plants undergo photosynthesis during daytime. And now coming to this diagrams. So questions have been asked from this diagrams too. One more thing. There's an MCQ, possible MCQ asked many times from this part is like, which is the source of oxygen released during photosynthesis? So you can see that oxygen is released during photosynthesis. So where does this oxygen come from? Is what donates this or what contributes this oxygen? Is that carbon dioxide or water? The answer is water. So the second point here, you see that splitting of water molecules into hydrogen and oxygen. So this oxygen is released out. Okay, that's what we breathe. So the answer is water. And the splitting up process occurs in the presence of light. Now this diagram, in many question papers we can see this diagram. So this diagram actually shows the upper epidermis with waxy cuticle, lower epidermis and stomata, and you can find the stomata is widely distributed. There's only one stomata shown here, but that represents that stomata are widely distributed in the lower epidermis. Then you can see this some beaded structures, and those beaded structures are represented as chloroplasts. And if you just closely observe, in either epidermis, like upper or lower epidermis, you cannot find chloroplast. Chloroplast is found only in the palisade and spongy cells, that is the mesophyll cells, the cells in between. But there are chloroplasts in the guard cells of stomata. So question can be asked like this: name the cell, only cell in the epidermis with chloroplast in it. It is guard cell, okay? And stomata is distributed more in the lower epidermis. So these are all MCQs, possible MCQs from this part. Now comes the next activity. So this activity demonstrates chlorophyll is essential for photosynthesis. This is also a possible question. And here some questions, some split up questions can be asked, like here. Why do we keep the plant in dark room for 3 days? So that all starch can be used up, that is to de-starch it. Then we keep it only for 6 hours, that is we give time only to prepare the starch, like prepare the starch, its conversion, not its transportation. And then here we immerse it in a beaker containing alcohol, and we boil it. Double boiling is done here. And why do we do that? Or sometimes they'll ask, like, what happens to the color of leaf, or what is the color of the solution? Solution here meant the alcohol solution. So the alcohol solution turns green. So we do that, or we boil it in alcohol to decolorize. Only then we could differentiate the color. Okay. So the questions can be asked in various ways, but so that you should understand entirely what the experiment stands for. And coming to the next part, so these points, we have seen in assertion reasoning parts. Massive amounts of gaseous exchange takes place in the leaves through the spores for the purpose of photosynthesis. So we have already said that photosynthesis, during photosynthesis, carbon dioxide is essential, and it's taken in through the pore, right? So massive amounts of gaseous exchange, and not only that, even during respiration it happens. But it is important to note here that exchange of gases occurs across the surface of stem, root and leaves as well. So it can even happen through stem, roots and leaf, not only through stomata. That's a point. And now opening and closing of stomata. So that takes place with the help of guard cells. So why do the guard cells open or close? It is by the water movement. Okay, so that can be asked as an MCQ there. Movement of water. Water enters or exits, and that determines the closing or the turgid and flaccid, or swelling up and shrinking of the guard cells. So process of opening and closing of stomata is very important, and you should mention that it is due to the movement of water. That is important. And now coming to the next experiment. This experiment is to prove carbon dioxide is essential for photosynthesis. So whenever an experimental setup is given, and if you find KOH there, definitely remember that it is something related with carbon dioxide. So in this 10th, you have related to KOH and carbon dioxide is photosynthesis process. So if they keep KOH here, it means in that setup, carbon dioxide is made not available to the plants, and the other plant, carbon dioxide is made available. Okay. So here one more possible objective is here. An MCQ can be asked, or an assertion reasoning can be asked here. Use of Vaseline to seal the bottom of the jar. Why do we use Vaseline? To keep the setup airtight, because we are looking for the carbon dioxide, action of carbon dioxide, so no entry or exit of air must happen. So to keep it airtight, we use Vaseline to seal. Now here they haven't mentioned about an experiment that demonstrates sunlight is essential for photosynthesis. It's so simple, you might have already learned it, like covering a part of leaf with a black paper and conducting the same kind of experiment, so that we stop the availability of sunlight only to that part, that restricted area or the area covered with the black paper, and then continue with the starch test. But the questions, many times the questions can come like this, where all the mixed things, all the things can be asked. Here you can just see they have given two setup, leaf A and leaf B. In leaf A, they have taken KOH solution, and it's just this part of the leaf is within the cork. So this part of the leaf is within the cork. It will not receive sunlight, and this part of the leaf also within the cork, it will not receive sunlight. So this region, so this is a glass beaker, so definitely it's getting light, but here carbon dioxide is not available. And here in the case, this glass jar is covered with a black cloth, so sunlight is not available here. So carbon dioxide is not made available here, or they have stopped the supply of carbon dioxide here, and here the entry of light is prohibited. So in that case, which leaf will show this, indicate the presence of stars? Actually, neither of the leaves show, because in this case carbon dioxide is not available, and in this case light is not available. So this kind of question shall be asked. So again, you have to think, reason it, and write. And now we have already spoken about the raw materials required, like carbon dioxide, water, and light. Now, other than that, what else is required for the plant growth? The other materials like nitrogen, phosphorus, iron, and magnesium are taken up from the soil, and they are very important. And see, before that, the availability of carbon dioxide and water, their sources can be asked. Carbon dioxide is taken from the atmosphere, and water is from the soil. Okay. Now, nitrogen is essential. This is one of the important MCQs. Nitrogen is essential element used in the synthesis of proteins and other compounds. And also the question can be asked, like, how is nitrogen made available to the plants? So this is taken up in the form of inorganic nitrates or nitrites, or it is taken up as organic compounds which have been prepared by bacteria from the atmospheric nitrogen. So this part is important. Now coming to heterotrophic nutrition. Again, differentiation is possible. So here we learn three types of nutrition, like saprophytic or saprotrophic, then parasitic, and holozoic. So three different types of nutrition we learn here. So the difference comes like this. Some organisms break down the food material outside the body and then absorb it. That is saprophytic, and their examples are here. Whereas holozoic, they take in the whole material and break it down inside their bodies. They are holozoic nutrition. Example, amoeba, human. We take it and then digest. And parasitic nutrition, some other organisms derive nutrition from plants or animals, from the other plants or animals without killing them. And examples of plant parasite and animal parasite. Plant parasite, cuscuta or amarbel, and animal parasite like ticks, lice, leeches, and tapeworms. Now, holozoic nutrition in unicellular organisms, two examples are quoted here. One is nutrition in amoeba, and the other one is nutrition in paramecium. So whenever you study like this, whenever you study these kind of concepts, always study in the form of differentiation. So you'll get maximum points there. So here also there's a possibility of differentiation question. So if it is differentiation question, you have to write the mode first. In amoeba, both of them undergo holozoic nutrition. So that is a common point. What special features do they have, or unique features do they have? Like amoeba have temporary finger-like extensions called pseudopodia, and using that they use. Whereas paramecium, they have a definite shape and they have a specific spot, and the food is entered only through that spot. And here food vacuole is formed. Now they have a gullet actually here, so you can mention this food vacuole along with that point. Then pseudopodia is also used for the movement, and here by the movement of cilia. Okay. Then coming to nutrition in human beings also, it is holozoic nutrition. So you must go through the structure of alimentary canal and the digestive glands. Alimentary canal is the canal that starts from the stomach region to the end here. So it's like a tube-like canal. Small intestine, then large intestine, and opening it in anus. So this is a canal, and the accessory glands together forms the digestive system. So here I'm just focusing only on the important points. Okay, here salivary glands secrete saliva, and the enzyme in it, salivary amylase, and the function of salivary amylase. It converts or breaks down starch, which is a complex molecule, to a simple sugar. Then, so before that, here they talks about enzymes, so biocatalysts or biological catalysts. Okay, remember that term. Then here always goes with the functions, the structure, function, secretion, functions. So salivary glands in mouth, saliva, secretion. Then coming to the gut, before coming to the gut, through the esophagus, the peristaltic movement is important. Actually, peristaltic movement is not found only in the esophagus part, it is found throughout the gut, okay, all along the gut. Now then coming to the stomach, muscular walls of the stomach. Once this question been asked, the muscular walls of the stomach help in mixing the food thoroughly with more digestive juices, that it keeps churning. Okay, so the functions of muscular wall of stomach was once asked question. And now functions of gastric enzymes. Remember, gastric enzymes refer to the enzymes found in the stomach. And so the question will be asked in this way. See here, see A, B, C. Gastric glands present in walls of the stomach secretes pepsin. So the action of pepsin is protein digesting enzyme. And the second one is HCl. HCl creates an acidic medium which facilitates the action of enzyme pepsin, and also it can kill harmful bacteria. So actually HCl carries, you can mention three functions for HCl, that is activating pepsin, pepsinogen to pepsin, then creates an acidic medium and kills bacteria. And coming to the last one, that is mucus, it protects the inner lining of the stomach. So this is one of the way the question can be asked, one possible way for asking this question. And I'm coming to the small intestine. So before that, here a mentioning of sphincter muscle comes here. This can be asked in assertion reasoning question. That is, the exit of food from stomach is regulated by a sphincter muscle. So sphincter muscle acts as a regulatory part. Then longest part of the alimentary canal is small intestine, and this is one of the frequently asked questions, like small intestine of herbivores is longer than that of carnivores. That is for the digestion of cellulose. So cellulose, the term cellulose digestion must be mentioned there. That is the keyword of that answer. The answer can be asked, the question can be asked in the other way around. Like carnivores, it is shorter when compared to herbivores. That also can be asked. Or sometimes they'll mention a herbivore and a carnivore, like a deer or a deer and a lion or a tiger. So, likewise. But you should understand that this part is the answer. Now name the site of complete digestion. Again an MCQ, important MCQ, small intestine. And you remember, name the site for this where the digestion begins. Remember, it's mouth, okay, because salivary amylase is the one which starts the digestion. So digestion starts at mouth. Now this region talks about the secretion of bile and the enzymes. So actually bile juice do not have enzymes, but they play an important role in digestion because they make the medium alkaline, as actually the medium was acidic created by HCl. Pancreatic enzymes cannot act on it, so bile makes it alkaline, and bile juice acts on it and it emulsifies the fat. Emulsification means breaking the larger globules into smaller ones. And here they haven't mentioned about the gallbladder, so it is there in the diagram, and questions also been asked about gallbladder. It's a place where the temporarily bile juice is stored. Bile juice is stored temporarily here. Okay, so it is mentioned in the diagram, so you may get a question. Gallbladder. Then coming to pancreas, pancreas secretes juices, and here you have mentioned you have been given with trypsin and lipase. Pancreatic trypsin and pancreatic lipase and pancreatic amylase is also there, so keep that in mind too. Then coming to small intestine. In small intestine also the digestion happens. Actually digestion gets completed in small intestine. There is a small contribution of small intestine too. They also contain glands which secrete intestinal juice. And finally, the complex food material is broken down into simpler ones. So what is the end product of digestion? Like proteins are broken into amino acids, complex carbohydrates are broken into glucose, and fats are broken into fatty acids and glycerol. Now next process, so digestion is completed. Now coming to absorption. Absorption is again carried out in the small intestine with the help of intestinal villi. So intestinal villi are finger-like projections which increase the surface area of absorption. And villi are richly supplied with blood vessels which take the absorbed food. So here the question can be asked like design of villi that increases absorption. And see here finger-like projections. So they increase the surface area of absorption, and they are richly supplied with blood vessels so that the absorbed food can be taken. And the last sentence actually deals with assimilation. Assimilation means how these simpler food substances are used by our body. So that is assimilation. Finally, the unabsorbed food. See, the unabsorbed food is sent into large intestine. So what is the function of large intestine? Where its wall absorb more water from this material. So that is very important. The function of large intestine, absorption of water, and the exit of material, this waste material is digestion, and that happens through the anus, and this is again regulated by the anal sphincter. This can be an MCQ. Okay, now we'll move on to the next one. So these questions we have already discussed over there, so I'm not wasting the time on that. Coming to respiration, so you see the figure. This figure actually talks about the evolution of carbon dioxide during respiration, and they really meant the comparison of amount of carbon dioxide in air with the carbon dioxide, like amount of carbon dioxide in our breath. Like see, in this test tube they have taken lime water, and using this fish curry they have pumped the atmospheric air into it, and here in this second setup a person is blowing air into it. So they are comparing like how fast the lime water turns milky. In this case, in the case 1, lime water turns milky very slow because the amount of carbon dioxide in the air is comparatively very less, and the amount of carbon dioxide in our breath is comparatively higher. So it turns lime water turns milky faster. So that's what this experiment talks about. Now here in this chapter, in this paragraph, first they talk about the definition for respiration. So the sentence given here is not in the form of a definition, but you have to reframe it. So the sentence given here is like the food material taken in during the process of nutrition is used in cells to provide energy for various life processes. So this breaking down or this utilization of food material to provide energy for various life processes is known as respiration. And here in this paragraph they talk about two types of respiration, like aerobic and anaerobic respiration. Again, it can be asked in the form of differentiation. So if it is asked in the form of differentiation, you can write point wise. So first, again, as I said, the first point can be the definition. So in the case of aerobic respiration, so this is the first point, breaking down of glucose in the presence of oxygen. So that can be the first point in aerobic. Whereas in the anaerobic respiration, without involving oxygen, that is, do not involve oxygen. So that's the first point in anaerobic respiration. Now coming to the second point, here you can talk about the end products. In the aerobic respiration, the end products are carbon dioxide and water, whereas anaerobic respiration, end products are ethanol and carbon dioxide. And then comes in aerobic respiration, the second step onwards, that is the breaking down of pyruvate using oxygen takes place in mitochondria, whereas in anaerobic respiration it takes place, the whole process occurs in cytoplasm. Then few more differences comes here, here the release of energy in aerobic process is a lot greater than in anaerobic process. So you can write it in the comparative form. Release of energy is greater, and in anaerobic process it is comparatively lesser. Okay, so this diagrammatic representation is very schematic representation is very important. So if you learn this only the schema, you will be able to answer any questions related to this respiration. So here you'll just see the schema. Glucose, it's a 6 carbon molecule. So the first step here is common process. So that's one of the questions which I mentioned here. That is glycolysis, that is breaking down of glucose. It's a common pathway for both aerobic and anaerobic process. So this is one of the important MCQ, okay? So the first process is a common one. You can see that is common. Then comes the 3 types. So it occurs in cytoplasm. So where does it occur? That it's also one of the frequently asked questions. Now here 6 carbon molecule is broken down into 2 molecules of pyruvate, each with 3 carbon, and a small amount of energy is released here. So most of the children ignore this part. A small amount of energy is released in the first step itself. Then it gets differentiated, like if it is anaerobic, that is in the absence of oxygen, the end products are ethanol, carbon dioxide and energy. So here it is actually incomplete oxidation. The oxidation process is incomplete, okay? So that's why ethanol is there. And this one is aerobic process, that is in the presence of oxygen. It occurs in mitochondria. Here it is completely oxidated. So complete oxidation. So there is only the release of water and energy too here. Now, in the lack of oxygen, okay, so there is always a confusion for the students, like what's the difference between aerobic in yeast and aerobic in muscle cells? Actually, in yeast, oxygen is available, but they don't take it. In anaerobic in muscle cells, we use the word lack of oxygen, okay? So though in the question they have not mentioned as muscle cells, if they have mentioned as lack of oxygen, you should understand that it is the respiration that is taking place in muscle cells. So the products are lactic acid and energy. The location or the site of occurrence is muscle cells. And here lactic acid is a 3 carbon compound, and this 3 carbon compound, since it's an acid, accumulation of it causes cramps, and this cramps can be relieved by massaging with hot water. So that hot water massage will improve our blood circulation, and when more blood, more oxygen, so their complete oxidation takes place and carbon dioxide is released later. Okay, that's how the muscle pain is relieved. Now the overall product of respiration is target, like what we expect is energy. So that energy is immediately converted into the currency, which we call as chemical currency or chemical form of energy. We call it as ATP. Expansion of ATP is adenosine triphosphate. And now how ATP is used up? ATP is used, or where do we use energy? So it is not only while making physical movements, all the life processes, all the internal processes we require, even all the molecular movements we could say we require energy. So whenever there is a requirement, ATP is broken down, giving rise to a fixed amount of energy, which can drive the endothermic reactions taking place in the cell. Why have they mentioned as endothermic reactions? Because the reactions which take energy use heat energy. Okay. And please go through this too. That can also be asked, like you don't expect directly the question like this, but in any indirect form this question shall be asked. Okay. Now this is also an MCQ. Plants exchange gases through stomata, that we have already discussed it, and large intercellular spaces ensure that all cells are in contact with air. So excuse me, I'm moving back to the figure of leaf, where they have shown the air spaces. So since there are more air spaces, when the air enters in, it gets in direct contact with all cells, so that through the cells it can diffuse into, because there is no special organ in the case of plants. Exchange of gases occurs by simple diffusion. So the gases must be made available to all cells, and that is facilitated, or that is enhanced, by the presence of air spaces. And this part again, the question, the statement of the framing of question gets confusing. The answer is not that tough. The question what they ask is the factors on which the direction of gaseous exchange depends. That is, how much gas is entering in, or how much carbon dioxide is entering in, or when does the carbon dioxide enter, when do the oxygen enter. So that exchange depends upon the external environmental factors, how much carbon dioxide is entering, or at what time carbon dioxide enters. Usually we say that during daytime photosynthesis occurs, so carbon dioxide is taken in. Do you think that plants respire during daytime? Yes, they do, but the amount of carbon dioxide released out is negligible. But during night, there is no carbon dioxide intake because there is no use. Since there is no light, it's of no use. So they don't take carbon dioxide at night, and they take oxygen at night and release carbon dioxide. So this is a core concept of that part. The direction of diffusion depends upon the environmental conditions and the requirements of the plants. Please get familiarized to these statements. I'm not asking you to learn it by heart, but just get familiarized, because most of the times the questions will be asked using the same statement in the textbook. So you might have learned the concept, but you would not have got familiar with the statements given in the textbook. So that makes you getting confused with the questions. So the direction of diffusion depends upon the environmental conditions and the requirements of the plants. Justify. Yeah. At night, what is the environmental condition at night? When there is no photosynthesis, because there is no light. Carbon dioxide elimination is the major exchange. So there is no necessity for carbon dioxide, no use of taking carbon dioxide. So the respiration process is more. So elimination of carbon dioxide is a major exchange activity. During the daytime, carbon dioxide generated during respiration is used up for photosynthesis. During daytime also, respiration occur, but it is used up during the photosynthesis, because immediately the carbon dioxide is taken in. So that's what I mentioned, that the amount of carbon dioxide released during respiration during the daytime is negligible. Okay, so here in that case oxygen release is a major event at this time. Okay, so we have already mentioned that the terrestrial animals can breathe the oxygen in the atmosphere, and the aquatic animals take the oxygen dissolved in water. So the oxygen is fairly low in water when compared to the amount of oxygen in air, so that the breathing in aquatic animals is much faster. They need to take more oxygen intake should be more. Okay, so that's one of the question here. So this question shall be asked in a different way. So you can see the in-text question here. What advantage over an aquatic organism does a terrestrial organism have with regard to obtaining oxygen for respiration? So again, here comes the same. In water, the amount of oxygen is comparatively low, and it is dissolved oxygen, whereas in the atmosphere, like the terrestrial organisms can get it from the atmosphere, where it is comparatively high. Okay. The question can also be asked like rate of breathing in aquatic organism is much faster than in terrestrial organisms. Why? So question also can be asked in that way. And this part, it's not that important, but just go through it. Fishes take in water through their mouth and force it past the gills. So how do they breathe in? They take water through their mouth and pass out through gills. In the gills, the dissolved oxygen is taken up by the blood vessels. The blood through blood vessels. Now here talks about the structure of respiratory system. So generally they say how a respiratory system, complex respiratory system is designed to facilitate the respiration, process of respiration. So the first thing is the surface area. So here in the case of human beings, we have lungs. Inside the lungs there are numerous alveoli. So that increases the surface area, which is in contact with oxygen-rich atmosphere. That is, we breathe the air in, oxygen-rich air in, so that air comes in contact with that many number of alveoli, and the exchange of oxygen and carbon dioxide has to take place across the surface. So in the alveoli, that exchange of oxygen and carbon dioxide occurs. And secondly, how that exchange is occurring, because the alveoli, the surface of alveoli is very fine and delicate, and also there will be a lot of blood vessels. So this will facilitate the exchange of gases, and in order to protect the surface, it is usually placed within the body. So that is again important. Alveoli are not exposed out, it is placed within the body, and so that we have to have air passages like the windpipe, trachea, bronchi, bronchioles and all. So this is how a respiratory system is designed for respiration in higher organisms, that is the terrestrial organisms. Now this part we have already learned in our lower classes, like why are we not advised to breathe through mouth? It's not advisable. It's always better to breathe through our nostrils, because our nostrils have certain structures that can filter the air. So hair, fine hairs as well as the mucus. Okay. Now this part is important. Actually, we said about the air passages here. These air passages must not collapse, so that it provides or it ensures continuous supply flow of air. So to make sure that those air passages do not collapse, it is made of rings of cartilage, so it will not collapse. Okay, so I always ask my children to compare the air passage with the draining out tube of a wash basin or washing machine. You can see the draining out tube. It is not that thick, but it has like circular lines. So that's how our trachea appears to be. Okay, you can compare it. Keep that in your mind so that it will not collapse. So it ensures the continuous flow of air. Now I've mentioned here as the order of passage of air. It means that the order, like air enters through nasal passage, it passes through pharynx, larynx, trachea, then bronchi, bronchioles, and enters into alveoli. So this order sequence is important. Even all other labels are important, must go through the diagram. Okay. Now again here they talks about the design of alveoli. How is alveoli modified to facilitate gaseous exchange? Because the exact process of gaseous exchange occurs in the alveoli. So we said already that alveoli has thin membrane, and also they have extensive network of blood vessels. There's quite a lot of blood vessels are there. Then next part, changes that taking place during inhalation. So only that changes occurring during inhalation is mentioned here, not exhalation. So let's see here, when we breathe in, we lift our ribs, flatten our diaphragm, our chest cavity becomes large as a result. Because of this, air sucked into the lungs and fills the expanded alveoli. So here they talks only about inhalation. Exhalation part also can be asked. So the reverse of this is exhalation. Reverse in the sense, our lips are when we breathe in, the ribs. When we breathe out, the ribs, what happens to the ribs? It moves, it closes inwards, diaphragm gets dome-shaped, chest cavity becomes smaller, and because of this, air is released from the lungs. Okay. Now, here this part is the exchange of gases in alveoli. Important. How exchange of gases occur? Carbon dioxide is taken from the rest of the body. It is brought to the alveoli with the blood vessels, and the oxygen in the air which we breathe in enters into the alveoli, and carbon dioxide comes out. Then comes the residual volume. So residual volume is a certain amount of air that always remains in our lungs. So when we exhale out, we don't completely exhale out. A certain volume of air remains in our lungs always. So that is called as the residual volume. So how this help us? Two questions can be asked. One is, breathing is rhythmic where respiration is a continuous process. Breathing is rhythmic means we just inhale, exhale. So certain contraction and expansion of certain muscles happen. Okay. So it is contraction, relaxation, contraction, relaxation. So there's a pause in between. It's a rhythmic process. It's not a continuous process. Whereas respiration occurs in the cellular level. So in the cellular level, it is continuous process. So there should be a continuous supply of oxygen. So here, there is a sufficient time for oxygen to be absorbed from the carbon dioxide to be released. So when breathing happens, oxygen enter, alveoli takes in, like blood vessel takes in, and blood has to carry to the cells, right? So during that rhythmic process, there can be a gap. So that gap is filled by the air present in the lungs as the residual volume. So that part is important. Then comes about the respiratory pigment. Remember, respiratory pigment is hemoglobin. Do you remember what the photosynthetic pigment is? Yes, it's chlorophyll. And this respiratory pigment, that is hemoglobin, is the one which carries the oxygen because they have higher affinity towards oxygen, whereas most of the carbon dioxide is carried through plasma. It is soluble, since it is soluble in water, it is carried through plasma. So this part, it is how is oxygen and carbon dioxide carried or transported. Okay. Most of these questions we have already discussed. Coming to transportation, in the transportation region, see this part whole talks about the circulation of blood. I suggest at this last moment, because it's high time, this last moment, rather than learning this whole paragraph, you can just refer the schema, and by referring the schema you can learn the blood vessels and its circulation. Okay, the 4 chambers, the blood vessels entering and exiting, and where does it carry the blood to, where it brings the blood from. Okay, so this schema will help you. After learning the schema, you can go to the structure of the heart. And here in this part, three important questions come. One, why is it necessary to separate oxygenated and deoxygenated blood? So very important question, frequently asked question. Here it is like highly efficient supply of oxygen is required in the case of mammals and birds, because we maintain our body temperature according to the changes in the external temperature. That's why we are called as warm-blooded animals. Whereas reptiles, amphibians, fishes, they are all cold-blooded animals. They do not require much energy because there is no regulation of body temperature. Okay, in that case, some mixing of oxygenated and deoxygenated blood is okay with them. Then in the case of fishes, single circulation in fishes, because they have only 2 chambers, and so only once the blood enters into the heart and comes out, so it is single circulation. Whereas in the case of vertebrates like mammals and aves like birds, double circulation. One is pulmonary circulation, so this is pulmonary circulation because it is concerned with lungs. It is taken to lungs, oxygenated and brought back from the lungs. This is systemic, where the deoxygenated is collected and oxygenated is brought back. Okay, so this is systemic circulation. These 2 terms are not there in your textbook, but still get familiarized with it. So that's why it's called as double circulation. Now coming to this part, blood pressure, it is really important. You have definition of blood pressure. The force that blood exerts against the wall of a vessel is called blood pressure. And now it is much greater in arteries than in veins. That's a known concept because it comes in the differentiation of arteries and veins. Now systolic and diastolic pressure. Systolic pressure is the pressure we calculate at the time of ventricular systole. Systole is contraction and ventricular diastole is relaxation. Okay, so that is systolic and diastolic pressure. And normal blood pressure of average blood pressure of a normal human being is 120/80 mm of Hg, where this 120 refers to the systolic pressure and 80 refers to the diastolic pressure. So systolic pressure, we have already mentioned that is due to the contraction, so the pressure will be more there. Ventricular contraction, where the blood is carried to whole body. Okay, whole body. Then now blood pressure is measured with an instrument called sphygmomanometer. That is also important. Then the next question I've mentioned here is about blood pressure, its reasons, its consequences, all comes in this part. Okay, reason, it's also called hypertension caused by the constriction of arterioles, results in increased resistance to blood flow. And its consequences, it can lead to the rupture of artery and internal bleeding. Now coming to difference in arteries and veins. So this part, you have the differentiation between arteries and veins. You can write the differentiation, their structure, the type of blood they carry, either oxygenated or deoxygenated. They carry from the blood, from the heart or to the heart. Then do they have valves? They do not have valves. How about the pressure in them? So all these points can be written there. Now coming to capillaries. Here an important MCQ in this part is like exchange of material between blood and surrounding cells takes place across this thin wall. So where does this exchange of material occur? That is in capillaries. Any exchange of material can be digested food or the excretory waste materials or the oxygen, like gases. Any exchange of things happen at the capillary level. And this is also another important MCQ, like capillaries join together to form veins. Okay. Capillaries join together to form veins that convey the blood away from the organ or tissue. Like it's like arteries divide into capillaries, arterioles, capillaries, and these capillaries unite to form veins. So arterioles and venules is the right term, but we can write as arteries and veins here. Okay, arteries split into capillaries, so this is the capillary form, and they rejoin to form veins. Blood platelets, you all know that blood platelets are the one that prevents the clotting of blood. So here the question comes like, what happens if there is leakage in blood vessels and how can it be prevented? How it is prevented? Naturally. So leakage, if it happens, what will be the consequence? Loss of pressure, so that it reduces the efficiency of pumping system. And to avoid this, blood has platelet cells which circulate around the body and plug these leaks by helping to clot the blood at the points of the injury. Right. Now, coming to lymph. In lymph, 3 questions can be asked, like how is lymph formed? Then the differentiation between the plasma of blood and lymph, and the functions of lymph. So how is lymph formed? Till this, or what is lymph? And the differences between the plasma of blood and lymph is like, in blood it is colored because it has RBC and it has more protein, whereas lymph is colorless, contains less protein. And then the functions, it carries digested and absorbed fat from intestine and drains excess fluid from extracellular space back into the blood. And here one more question is possible here, like where do these lymph vessels drain? They drain actually into lymphatic lymphatic capillaries drain into lymph vessels, like lymphatic system, and later it is opened into the larger veins. Okay, so this is also an important MCQ. Now transportation in plants. Transportation in plants, there are 2 things to be carried out in plants. One is water transportation and the other one is food transportation. So water transportation is carried out through xylem, and that process is called ascent of sap. Ascent of sap is always unidirectional, that's only upward movement. And ascent of sap, so these, when the question is asked you to describe the transportation of water through xylem, these points are very important. That is actively taking up of iron from the soil. The roots will take actively means by active transport. And when there is an increase in iron intake, there will be a difference in their osmotic pressure, and that difference will cause the water entry, and so that now the water has entered into the root, and then by the suction pressure of the transpiration, water will be moved upwards. So that suction pressure that keeps that steady column upwards is by the transpiration. So that comes here. What suction pressure causes the upward pull of water? It is transpiration. And here this first 4 lines talks about the advantages or uses, functions of transpiration, or significance of transpiration, you can say. That is, it helps in the absorption and upward movement of water and minerals. Also, it maintains the temperature. So you'll get a cooling effect because of this. And if the question is asked like transpiration is a necessary evil, you have to mention for the necessity of transpiration, because this both points as necessity, and evilness is nothing but it's obviously loss of water, right? So it is an evil. The water is lost. Then During daytime, the steady column of water is maintained by the suction pressure created by transpiration. And what about night? So at night, root pressure helps in the transport of water. So this is yet another MCQ. Now, transportation of food. So it's very important. It happens through phloem, and not only food, prepared food, but also amino acids and even other substances, and where this food is taken. Next question, where is it taken? Where they are taken to? They are taken to storage organs of roots, fruits, seeds, and to growing organs. And the cells or the structures involved in this is phloem, especially phloem, and in phloem, the phloem elements like sieve tubes and companion cells both take place, and it occurs by like upward and downward direction. So I think you'll get some differentiation points like in transpiration of water through xylem and transpiration of food through phloem. Transpiration of food through phloem is called as translocation, and the previous process is called as ascent of sap. So ascent of sap is always unidirectional, that's only upward, and this translocation occur in both upward and downward direction. So the process of this translocation is very important. Okay. So in water transport, there is no expense of energy actually. Only when there is an active transport of ion in the root, a small amount of energy is used up. But there, only to increase the osmotic pressure it happens. And here, while the transportation of food, it happens at the expense of energy. So it is achieved by utilizing energy. So it's one of the major difference between the translocation and ascent of sap. So this part is important, and again here they have mentioned where it is taken to. Okay. Most of all these questions we have already discussed, so I'm not wasting time on it. Let's come to excretion. Definition of excretion, that is the biological processes involved in the removal of harmful metabolic waste, that is nitrogenous waste. So what are the metabolic waste? Major waste are nitrogenous materials. Okay. So as we said in the beginning of the chapter, multicellular organisms require complex organs, whereas unicellular organisms can achieve this by the simple diffusion. Now, parts of excretory system, you must know at least to label kidney, ureter, urinary bladder and urethra. The other parts are also important. I'm not saying that they're less important, but you must at least know these four regions. Okay, you must know to draw this too. Now, how is urine produced? So urine produced, the answer starts here. The basic filtration unit in kidney is nephron. So the term nephron is not mentioned in the descriptive area here, but it's mentioned in the diagram. So you must know that term. And diagram of nephron is also important. You need not draw this much coils. You can draw it in an easy method. And the name of the regions which you must know is Bowman's capsule, and glomerulus is mentioned here. It is the blood capillary, tuft of blood capillary, so it's called as glomerulus. And once the filtration starts, we call it as initial filtrate. So initial filtrate can be asked as a separate question. What is the composition? It has glucose, amino acids, salts and major amount of water. And this water is reabsorbed, that is tubular reabsorption. So you have first one, initial filtration, then selective reabsorption, and at certain areas there will be tubular secretion. So here only two things you are learning, like initial filtration, second one selective reabsorption. Then finally the urine formed is collected in the urinary bladder. And here two more questions which you get. As I said, one is initial filtrate and the second one is the quantity of urine is regulated by how much amount of urine gets out of our body. So what regulates that? Two factors they have mentioned here. One is like the amount of water reabsorbed. How much water is reabsorbed, selectively reabsorbed. Second thing, on how much of dissolved waste is there to be excreted. So these are the two factors they have mentioned here. But one more hormonal factor is also there. So if you can keep that in your mind, please do remember a hormonal factor also has an impact in the quantity of urine formation. Okay. Then this last part, bladder is muscular, so it is under nervous control. Why? Because Once the bladder is full, it's muscular, so once it gets full, it sends messages to the nervous system saying that the bladder is full, it has to be emptied out. Okay, so that is when we get the urge to urinate. And coming to dialysis part, you'll get 2 questions. What's the major difference between the blood and the dialyzing fluid? Here, the fluid has the same osmotic pressure as blood, except that it is devoid of nitrogenous waste. So this is one of the possible question here. Then lately they asked the amount of urine released from our body is on an average of 1 or a liter or 2 a day, but the initial filtrate is about 180 liters. So how that is, what makes the difference? So it is only due to the tubular reabsorption, that is selective reabsorption. So that much water is reabsorbed. Okay. And now coming to excretion in plants, I have jotted down like 1, 2, 3, 4, 5, 6. All these 6 are the types of excretory waste materials produced in plants and the way they use to eliminate it out. All 6 points are important. Like oxygen. Oxygen is a waste product, though it is our life-saving process, but it is a waste product, not a waste product. Released during photosynthesis. Plants do not use it, so they release it out. So it can be considered as a waste product. Then oxygen, carbon dioxide too, during respiration. Then they can get rid of excess water by transpiration. Again, process of excretion. The excess water is sent out. So sometimes when the excess water is sent out, along with some waste material is also sent out. Then dead cells will have waste material. So old leaves, dead leaves falling down, carrying all the waste materials. Then which are stored in their cellular vacuoles, and resins and gums which are secreted, exuding out from the plants. They are also some of their excretory products, and plants also excrete some waste materials into the soil around them. Okay, so this is all about this chapter. Now, as I said earlier, remember, this is not the time to learn something new, but still, if you find something new, so whatever I've mentioned in this video contains all the possible but frequently asked questions. So I can assure you that if you go through all these questions, if you learn all these questions, definitely you can clear all the questions from the life processes. So if you have any doubts, any part to be clarified, you can just mention in the comments. We'll be back soon with the next chapter, the quick revision of the next chapter. Thank you. of organic substance, and